Hello and welcome to this Bioprocess International Ask the Expert webcast. I'm your host, Alex Nicolaitis, the webcast coordinator for Bioprocess International. Before we get started, just a couple of notes. This webcast is being recorded and will be made available for replay in the multimedia section of our website. We've muted the audio lines, but you're welcome to type in your questions for our speaker in the question and answer on your after the presentation. Begin the name, and I'll ask our speaker your questions. Your questions in the Q&A window will only be visible to myself and our speaker. So thank you for joining us today. It's my pleasure to introduce our speaker, Alla Zilberman, PhD, Vice President, Technical Marketing from Cygnus Technologies.
Thank you, Alex. Thank you and thank you everybody for attending Cygnus Technologies webinar. My name is Alla Zilberman. I'm responsible for technical marketing at Cygnus Technologies. Unfortunately, Dr. Jared Isaac could not present today due to his unexpected illness. In my presentation, I will focus on Cygnus antibody affinity extraction mass spectrometry method and go over several case studies demonstrating its utility for identification of immunoreactive host cell proteins to establish host cell protein antibody coverage. I will talk about HCPs enriched during process changes and HCPs co-purified with drug substance. Here is an overview of what we will cover today. First, I will introduce our HCP ELISA and just in general HCP ELISAs, and then the antibody affinity extraction method and how it can be combined with mass spectrometry for coverage analysis and HCP identification. I will then go over three different customer case studies. So in the late 90s and early 2000s, host cell proteins were considered as a process monitoring tool only. And it is not until some clinical trials were put on hold due to higher than expected rate of anti-CHO antibody development that we started to really focus on better methods for HCP detection, and specifically, we started to look at HCP immunoassays. Today, we know that there are many potentially problematic HCPs that may co-purify with final drug substances. These host cell proteins can affect drug product stability by degradation through proteases or through residual lipases causing degradation of such excipients as PS20 and PS80, which are often used in formulation buffers. Some HCPs can increase immunogenicity and impact drug efficacy. You may be familiar with some of those on this list, and there are quite a few publications out there. It seems like every few months we see a new publication about this HCP or that HCP implicated in certain either side effects or certain affecting product quality. And as of today, HCP ELISA continues to be the gold standard method for HCP detection and quantification. At Cygnus Technologies, we have been developing and providing HCP ELISA to the industry for over 25 years. This slide represents a typical Cygnus HCP ELISA, which is a sandwich ELISA. Anti-HCP polyclonal antibodies are immobilized on a plate. And in step one, HCP samples and HCP standards are added to the plate and incubated for 2 hours with HRP label detection antibodies. Then the plate is washed to remove any unbound reactants. In step two, TMB is added and the colorimetric change is read in 30 minutes. So it takes just over 2 hours and 30 minutes to run a typical Cygnus HCP ELISA. So here is, I would like to talk about here I will talk about the different advantages and disadvantages of HCP ELISA. ELISA continues to be the gold standard for process monitoring and product lot release. And there is a reason for that. One of the biggest reasons is that it can measure nanogram per mil levels of HCP in the presence of milligram per mil levels of product protein. If designed well, it has excellent sensitivity and selectivity. It does not require special expertise to run, and it can be easily transferred across an organization or to a CDMO across the world. Unfortunately, the HCP ELISA also has some limitations. At the end of the assay, you read the plate and the software reports a result for each sample. This result does not tell you anything about what HCPs are in the sample or which HCPs the assay reacts to. We also know that ELISA is a semi-quantitative measurement of a statistical sample of the host cell proteins in a sample. It does not actually measure nanogram per mil. In reality, it measures immunological equivalent relative to the HCP standards. Therefore, results are subject to change, sometimes significantly, with a change of reagents supporting that ELISA. And while HCP ELISA does not require a high level of expertise to run, it does require a high level of expertise to develop a broadly reactive HCP antibody and then turn that into an accurate and robust immunoassay. So none of those issues are lost on regulatory agencies. So it is required to use orthogonal methods to demonstrate that an HCP ELISA is fit for purpose of process monitoring and product lot release. So to qualify an HCP ELISA, one must show good dilution linearity across a range of samples from upstream harvest all the way to the final DS. It has to demonstrate accuracy by performing spike and recovery analysis experiments, precision, and determine the range of the assay. But there is one additional and important requirement for HCP ELISA, which sets it apart from other ELISAs. You have to prove that an HCP antibody supporting this ELISA has sufficient coverage and can detect a wide range of HCPs that are present in each bioprocess. So antibody coverage, so today we will focus on antibody coverage by antibody affinity extraction, coupled with 2D page and silver stain detection. We also will talk a lot about the mass spectrometry and how the assay and antibody can be truly characterized by performing antibody affinity extraction combined with mass spectrometry. So antibody affinity extraction, or AAE, it's a Cygnus coined term, but it is referenced as immunoaffinity chromatography in USP Chapter 1132. It has been used in over 300 projects to perform antibody coverage analysis for regulatory submissions. So in the coming slides, I will review the AAE method and its advantages over the more antiquated methods such as 2D Western blot. I will also show how AAE combined with mass spectrometry can be used for identification of HCPs in mock and null harvest, but most importantly, as well as product expressing harvest for coverage. AAE is also a very powerful tool for HCP identification in the presence of DS, or drug substances. This diagram outlines Cygnus AAE method. First, we immobilize HCP antibody on a resin and pack a small 1 ml column with this antibody derivatized and conditioned resin. Conditioned because we want to make sure that there is no unreacted sites that would not specifically bind HCPs. In this example, an aliquot of a harvest sample is saved and the remainder of the sample is run over the EE column. The immunoreactive host cell proteins are captured with immobilized polyclonal HCP ELISA antibodies. Then the column is extensively washed to remove non-reactive proteins and those proteins that may still be on the column due to non-specific protein-protein interaction. Obviously, we don't want to overestimate coverage. Finally, the immunoreactive proteins are eluted from the column, and then pre- and post-AE samples are analyzed either by LCMS or 2D page. And the results are reported either in gel format, as if we do 2D page, or we can, if we do mass spec, we can identify all the HCPs that are in the harvest, all the HCPs that reacted or immunoreactive HCPs, those are that antibody have reactivity to, and we also will know their molecular weight and PI, or isoelectric point. And because we know those parameters, we will be able to build 2D virtual page with mass spec data. In this slide, I show a typical workflow of MS-based HCP analytics. So instrumentation shown here is intended to be illustrative of the potential combinations that may be employed for HCP analytics. In general, samples are enriched for HCPs by AAE and then tryptic digested. Custom LC-MS methods are developed to the specific host cell line, drug substance, sample matrix, and purification process. After method development, samples are run by liquid chromatography mass spectrometry upon a high-resolution, accurate mass spectrometer. Mass spectrometry data are searched using bioinformatics software, and all data searches use Cygnus Technologies' proprietary curated proteomic databases that we curated with common species expression systems. So AAE was developed in response to the limitations of the 2D Western blot. 2D Western blot significantly underestimates the true reactivity of the ELISA antibodies. So because 2D pages run under reducing and alkylating conditions, this denaturing sample preparation destroys the native epitopes that the ELISA antibodies use to bind to the HCPs. Gel transfer to PVDF membrane can also dramatically affect results. Too long or short transfer times will impact the membrane penetration of high and low molecular weight proteins, leading to misrepresentation of immunoreactive proteins. PVDF membranes can also hide the binding epitopes of ELISA antibodies, and they can also trap secondary antibodies, leading to large non-specific spots. Lastly, exposure time of chemiluminescent or fluorescent signal influences final Western blot results. In contrast, AAE is run under native conditions similar to how the antibody immobilized in an ELISA plate wells react with HCPs in the samples or standards. AAE 2D page, of course, is an improvement over 2D Western blot, but gel artifacts from gel running and fixation are still limitations because you have to fix the gel, then you have to make sure that you rehydrate the gels and you compare the images. But thankfully, mass spectrometry does not have limitations. Also, one of the criticisms of the AAE was that, do you know that our method is specific? So here on the right, you can see two panels, A and B. So the panel A shows the chromatogram of the CHO antigen bound of the column with CHO antibody, and we load CHO antigen on the column, and you can see there's a big peak on the left, and then we do AE for 4 cycles, and for each cycle we can see 4 specific immunoreactive HCPs elutions. The panel B shows the same experiment, but when we load the CHO antigen on non-imbued gold IgG column, and you can see that virtually no HCPs bound and eluted of that column. So this shows the specificity of the AE method. So AE combined with mass spectrometry is a powerful and informative technique, as it allows identification of the HCPs that are reactive with HCP ELISA antibodies. So here we show the data for our F550 anti-CHO HCP polyclonal antibodies in a CHO harvest sample. The panel A shows the results by 2D Western blot. The gel on the left shows the pre-AE CHO harvest sample, which represents all HCPs in your null or null sample on your harvest sample, and the gel on the right shows the post-AE HCPs, and those are the proteins that are reactive with antibodies. This is our antibodies, so to speak, cover those HCPs. And you can see here that by 2D page, our coverage percentage is 86%. The column B shows in panel B, we have 2D virtual gel of the MS data with matching proteins in green. Those are the proteins that we found in both in the pre-AE sample and the post-AE sample. The dots shown in red are those proteins that our antibody do not cover. We miss them. It's virtually impossible to develop 100% coverage antibody. And then I would like you to pay attention to those black dots. And those black dots are the proteins that were enriched by AE. it means, and were detected by mass spec, it means that they were there before the AAE. They were just below the limit of detection of the method. And by AAE, we enriched them, and we know that our antibody reacts with those HCPs as well. So by mass spectrometry, you can see that coverage of our antibody is 97%. Another thing that I would like to point out here, and as we go through this presentation, is that that with AAE to the page, we just, any gel method just measures spots. We don't know anything about those proteins. But with mass spectrometry, we know exactly what HCPs our antibody reacts to and what HCPs are in your harvest. So in the past, when somebody would ask us, do you know if our antibody reacts to this protein or that HCP? We can say at the most, maybe. We really were guessing. We didn't have the exact, we didn't have the insightful, accurate results here. However, today, with mass spectrometry, we can confidently answer this question. In this example, in figure B, we show that our CHO-HCP antibody reacts with PLBL2 as evident from the 20-fold enrichment of this protein by CHO-HCP antibody AAE column. In figure C, the peptide spectrum used to positively identify PLBL2 is shown. And in table A, we show that our two CHO antibody from our older F550 kit and our current F550-1 kit. The green and yes column indicate that both F550 and F550-1 antibody cover all of the potentially problematic HCPs in the sample. The red and no columns indicate the absence of the HCPs. This table also shows top 40 problematic CHO HCPs and shows that our antibody reacts to those proteins. This is very powerful information that can help you in overall risk assessment that comes from the HCP and you incur during your process development. So we applied the same methodology to our HEP293 HCP antibody, where we have performed coverage analysis by 2D page Western blot, by AAE 2D page and by mass spectrometry. And you can see here that coverage by 2D page is 95%. By combining AAE with mass spec, we identified more proteins, both in the starting material, in the harvest, and immunoreactive proteins. And our overall coverage went to 95%, but it's still pretty close, 91 or 95%. I would like to point to bring your attention to the fact that at Cygnus, when we introduce new resupply antibody or resupply assay, ELISA, we always characterize our antibodies by two methods, by AAE 2D page and AAE mass spec. And here you can see one of our recent resupplies with E. coli, where we can also characterize our resupply E. coli antibody by those two methods. And you can see the coverage in the high 80s, 87 and 89% respectively. So it's all great. I mean, we are done with coverage here. Coverage is obviously very, very important to make sure that you are using correct ELISA for your process. But it is really those proteins that make it all the way to the final drug substance through a given purification process that are the most important proteins with respect to patient safety and drug efficacy and stability. So a major challenge for LC-MS-based identifications of HCPs and drug substances is that there can be more than 5 orders of magnitude difference in the concentration between HCPs and drug substances, which precludes the effective identification of low abundance HCPs. So we apply the same AAE mass spec-based workflow to identify HCPs that are being quantified by the HCP ELISA in the final DS or drug substance. So the same here, we save an aliquot of drug substance, and the remainder of the samples run over the AE column. The immunoreactive host cell proteins are captured with the immobilized polyclonal ELISA antibodies and are eluted from the column. We repeat the cycle with this step for 4 cycles, and then we have pre-AE sample and post-AE sample, which we analyze by LC-MS, and the results are reported as a list of all identified host cell proteins with their respective molecular weight and isoelectric points. So by subjecting drug substance sample to AE, we can deplete most of the drug substance and enriched HCPs, especially the low abundance HCP, which are reactive with the HCP antibody, and thus they're still quantified by corresponding ELISA. This slide further demonstrates my point. The relative abundance of DS and HCPs according to the extracted ion chromatograms were normalized to 1 and graphed as a stacked bar chart. Pre-AAE column on the left shows the drug substance heavy chain and in orange, and the drug substance light chain peptides in light blue, and the HCPs in assorted colors beneath. Post-AAE column or bar on the right shows that the relative abundance of the HCPs has been increased after AAE, and that of the drug substance heavy chain and light chain has decreased. So we can see much more in depth the profile, with much better definition, the profile of the HCPs in the final DS. Here we demonstrate this approach with 4 independent drug substances from 4 different client projects. In the top panel, we show a virtual 2D gel of 2 HCPs in a monoclonal antibody drug substance, and the DS proteins are shown in orange, and the matching HCPs are shown in green, and the AE enriched HCPs are shown in black. At the bottom table, you can see the number of HCPs identified in those 4 drug substance samples prior to enrichment. This is a straight tryptic digest, and after AE enrichment, and we can see that you can consistently identify more proteins. We also compared our antibody affinity extraction enrichment method and DS depletion with the method which is frequently used and by many mass spec groups at biopharma companies. It's called native digestion. Native digestion is an HCP enrichment technique that was developed by Huang et al. At Eli Lilly, and it was published in Analytical Chemistry in 2017. You can see the reference at the bottom of the slide. To benchmark the performance of AAE and native digestion, both methods were compared and were performed on 2 drug substances, drug substance 1 and drug substance 2. You can see that without enrichment, there were 16 and 27 HCPs in drug substance 1 and drug substance 2, respectively. In both cases, AE outperformed native digest in terms of number of proteins identified. AE enrichment resulted in 32 HCPs identified, while native digest identified 20 HCPs in drug substance 1. So likewise, AE enrichment resulted in 44 HCPs, while native digest sample preparation enabled identification of 33 HCPs in drug substance number 2. Additionally, AE led to the identification of 100% of all HCPs found in the unenriched samples, while native digest only captured a subset of those proteins. So all HCPs identified via native digest sample preparation method were also identified via AE sample preparation method, which further supports that both methods enrich for similar population of HCPs. But AE does this much more effectively. So next, I will go quickly through a few case studies. So in case study 1, we had an opportunity to compare 3 different coverage analysis methods. The client first performed coverage analysis by 2D Western blot analysis. A third party performed the 2D page with cyprofluorescent stain and calculated the antibody coverage to be 54%. Cygnus Technologies performed AE mass spec and AE 2D page on the same customer harvest sample, and AE 2D page showed antibody coverage of 89%. And a coverage by mass spec, 93%. This data demonstrates 3 very important points. First, antibody coverage is dependent upon detection methodology, with 2D Western blot being the least sensitive method. Second, both AE 2D page and AE mass spec are in high agreement. By 2D page shows protein spots on the gel, while LC-MS identifies unique protein. So the same protein can be represented by more than 1 spot, and you can see the number of spots in the start and null harvest by 2D page was 2,046, while in starting material by mass spec, it was 1,512 unique proteins. So this is very important. We are identifying unique proteins with mass spec, not gel spots. And the third point that I would like to make is that gel-based methods are highly dependent on the operator skill. The 2D page Cypress stain of the starting material in 2D Western coverage study shows 1,297 protein spots, while 2D page silver stain of the pre-AE sample of starting material shows 2,046 spots. Both stains have similar sensitivity, so we can attribute this difference really just to the skill and how the 2D page is run, because this is just the straight sample without any treatments, without the transfer to the membrane or without the AAE. So it's a starting material. So again, this is also a very important point. Next, I would like to point out a very important application for the AAE mass spec coverage analysis. So ideally, once the selected HCP ELISA method is qualified and validated as fit for its intended use of process monitoring and product load release, it is imperative to ensure that critical reagents remain the same for a given ELISA. Sometimes it's inevitable if the product is available for a long time on the market, or if you're using generic assay, sometimes the change is inevitable. And mass spec is uniquely positioned to really identify and characterize antibodies and whether the coverage remains the same. So here we compare the F550 antibody from our historic CHO HCP ELISA with our current HCP ELISA F550-1, and you can see that both antibody coverage is pretty high, around 97%. However, we also go one step further and we look at the overlap between those proteins that antibody react to. And so we can see that both antibodies react to 96% of the same protein. So this is a very powerful characterization that we always do when we do antibody resupply. The same was applied to our HEC293 antibody when we resupplied them in 2021. Again, this is the same type of data, and we have here the Venn diagram showing that the antibody are similar by 92% similarity, meaning that they recognize 92% of the same HEC293 HCPs. And then the final case study that I would like to show is that one study from one of our clients where they had two different processes. The process number one wasn't performed in 2022. The process number two was changed and performed in 2023. And in both cases, we showed and we demonstrated the coverage was pretty high, between 92 to 94 in 2022 and 96% in 2023. So this data served as a basis for the client to submit this process change to the regulatory agencies, and this data have been accepted by EMA. But what is important here is we compared the HCP profiles between the two samples and determined that they were 69% similar only. In figure A, we have the 2D virtual gel showing the overlay of the HCP presence in both samples. And as you can see, there's a high amount of green, which indicates the matching HCPs. But there's also red spots corresponding to only those HCPs that were found in the 2022 sample, and the blue spots corresponding to the 2023 sample. In figure B, we show that the overlap is only 69%. But what is important is that repeating the antibody coverage assessment was critical, as the process change resulted in 31 different HCPs. However, the coverage still was pretty high, and it means that the antibody are covering most of those HCPs, and the client can still use the same validated HCP ELISA to continue with manufacturing of their drug substance. So in conclusion, I would like to, in summary, I would like to provide those kind of take-home messages that, first of all, AAE mass spec generates accurate HCP antibody coverage data. we know valuable information about what HCPs are present in a given process and which are detected by the respective HCP antibodies. It allows us to shift the conversation from a simple question, what is the percentage coverage, to a much more insightful one. What specific HCPs does your HCP ELISA detect and quantifies? And this is extremely important. AAE mass spec also identifies and assesses reactivity of HCPs in process and drug substance samples. It helps you in assay selection, in risk assessment. It can be used to compare the reference standard to that similar. It can also lead to improvements in downstream purification processes. For the sake of the time, I wasn't able to show you a couple more case studies when we use AAE mass spec to troubleshoot HCP enrichment during downstream purification process or scale up, but we do have those data upon request. So in summary, AAE mass spec enables real data-driven decisions. So I would like to acknowledge and thank you all my colleagues at Cygnus Technologies that contributed to this presentation. And I thank you everybody for your attention.
Okay, great. And as a reminder to our audience, you can go ahead and type in your questions for Alla now. So our first question is, what is the sample volume requirement for coverage analysis by AAE with 2D page and AAE MS?
We typically ask for about 30 mils of the clarified harvest. It doesn't matter whether it's a null harvest or product expressing harvest. I know that a lot of clients do not have access to the null harvest, so this is not a problem at all. We can just take 25 to 30 mils of the product harvest, clarified product harvest, and perform coverage analysis on that sample.
And our next question is, what about those proteins that are not recognized by anti-HCP-AB? Will MS identify them?
Yes, it's a great question. Obviously, no method is without limitations, right? And so with mass spectrometry, yes, we will be able to identify HCPs in the starting material. So we will know whether this HCP is present in your harvest material for as long as it is above the limit of detection of mass spectrometry, which is 1 ppm for our instrument. Yeah. But it will miss, obviously, if your HCPs are at a very low concentration and the antibody do not recognize them, those will be missed. And I think this might be some 2-3% or yeah.
All right, and our next question is, if we perform coverage analysis by AAE with 2D page, do we still need to perform 2D WB?
Yeah, that's a good question. Sometimes there's some confusion that still exists out there. So if you performed AAE with 2D page silver strain, why would you need to do 2D Western blot? There's absolutely no reason to do 2D Western blot because what we have on our AAE column is anti-HCP antibody on which HCP ELISA is based. And what we do with AAE, we basically perform this immunological reaction between the HCPs in your sample and the antibody, HCP antibody. We perform it in a native biophysical environment, in exactly the same environment as we see in the ELISA plate when we add HCPs in the solution in the ELISA well. And this is where an antibody are immobilized in the well. So this is how interaction occurs. And basically we have the pre-AE sample, which is similar to the Western blot. It's basically the straight out harvest sample. And then the post-AE sample represents all immunoreactive HCPs. And only after that immunological reaction occurs, we actually do 2D page silver stain. At that point, we don't care whether the proteins are denatured because we already have two different gels. We have one gel with the starting material and a second gel with all immunoreactive HCPs, and we now can compare two images from the pre and post-AE silver stain gels, and we can have the assessment of the coverage. Again, with AAE 2D page silver stain, we only identify protein spots. We only count protein spots. We don't identify HTPs. That's why we are such in favor of the AAE mass spec, because it truly shows how the antibody are reactive, what HTPs are quantified by your ELISA. Yeah, but absolutely, you don't need to do 2D Western blot.
All right. Well, thank you, Alla, for that. It does look like we are running a bit short on time, however, so if we don't get to your questions, we'll be passing them along to Alla, who can follow up with you directly. So please go ahead and continue to type in any questions that you might have, and we'll get those passed along. The last question for the webcast is, do you provide top 40 problematic HCPs for each of the cell lines you can analyze by AAE-MS?
Yes, when we perform coverage analysis by AAE-MS, we always provide the list of all HCPs in your sample, list of all HCPs immunoreactive with antibody, and we have a separate table where we show top 40 problematic proteins for majority of the cell lines for which Cygnus provides HCP ELISAs.
All right, fantastic. Well, Alla, thank you so much for your time this morning.
Yeah, thank you everybody for attending our webcast.
Yeah, and the recorded version of this webcast will be available for on-demand viewing on our website. And as a registered attendee, you'll receive a follow-up email providing you with the direct link. We look forward to having you at future Bioprocess International Ask the Expert webcasts. Look for those announcements in your box. Goodbye.